Welcome back, everyone. Today we're diving into a paper that caught my eye. Structure language models for protein conformation generation.
Oh yeah, this one's exciting.
It is. And you know me, always fascinated by anything that can shake up how we think about proteins. And this one, well, it's all about predicting protein shapes. You know, all those crazy ways a protein can fold.
Right, right. Like, why is that such a big deal?
Well, think of it like this. Knowing how a protein folds is like having a cheat sheet to its function.
Oh, okay.
It's crucial for drug development, designing medicines that actually work.
Yeah, that makes sense. I mean, it's like imagine a protein is this super intricate origami creation.
Ooh, I like that.
It can fold in all sorts of ways, and every single fold, it affects how it works, how it interacts with other molecules.
Exactly. And traditionally, figuring out those folds, it's a pain.
Oh, tell me about it. It's super time-consuming. Computationally expensive, too, like trying to solve a massive 3D puzzle.
Ugh. Yeah, no thanks. But that's where this paper comes in with a potential solution. Structure language modeling, or SLM for short.
SLM, huh? So what's the gist?
Well, think of it like this. They treat protein structures like a language.
Wait, really? Proteins talking to us now?
Not quite talking, but more like we're deciphering their secret code. SLM translates those twists and turns into a protein vocabulary.
Okay, I'm intrigued. So how do we go from like a 3D protein to a vocabulary?
Good question. It's a two-step process.
Hit me with it.
First, they use something called a Discrete Variational Autoencoder, a DVAE.
DVAE. Okay, so what's that do?
It's like taking a huge image file and zipping it up. It makes the information way more manageable.
So like compressing a protein structure.
Exactly. And from that compressed information, we get our protein vocabulary.
Okay, so we've got the vocabulary. Now, how do we predict the shapes a protein can actually take?
That's step 2, conditional language modeling. This is where the protein's amino acid sequence comes in.
The building blocks of the protein, right?
You got it. It's like predicting the next word in a sentence based on what came before. But instead of words, we're predicting structural features.
So the algorithm's learning the grammar of protein folding.
Exactly. The paper talks about a specific way they do this called ESM Diff.
ESM Diff. Okay, another acronym.
I know. But this one builds on a powerful protein language model called ESM3. Like, think of ESM3 as the engine behind it all.
Got it. The powerhouse.
And by using a technique called masked discrete diffusion, ESM Diff gets really good at generating all those different protein shapes.
Okay, that's a lot of technical terms, but bottom line, does it actually work?
That's the exciting part. They tested it on a well-studied protein called BPTI.
BPTI, okay.
And SLM nailed it. It predicted even the really tricky, less common shapes of BPTI.
Wow, so it's not just solving the puzzle, but finding those hidden pieces, too.
Precisely. And it doesn't stop there. They also looked at how SLM handles those shape shifts that happen when proteins bind to other molecules.
Like when a drug binds to a protein.
Yep. They call them apophylo pairs. Basically, the protein's shape with and without a molecule attached.
Makes sense. So is this just theoretical, or are we talking real-world applications here?
Oh, this has practical implications for sure.
Yeah.
Especially for drug development. Knowing how a protein's shape changes when it binds to something is crucial.
Right. So if we can predict those changes more accurately.
Exactly. We could design more effective drugs that actually hit their target.
This is getting good. So how efficient is this whole SLM thing? I mean, we talked about traditional methods being a slog.
And this is where it gets really exciting. They found SLM to be significantly faster, like 20 to 100 times faster in some cases.
Whoa! Seriously, that's a game changer.
Right. It's like having a protein research superhighway. And that speed comes from how SLM works, that whole protein language thing, compressing the information.
So it bypasses those computational traffic jams.
Exactly. So we've got speed, accuracy, and it can even reveal those hidden protein shapes.
This is definitely a paper worth paying attention to.
Absolutely. But before we get too carried away, let's dive into those practical applications.
Okay, yeah, let's get down to the nitty-gritty. How's this going to change things in the real world?
Well, imagine being able to rapidly generate a whole library of possible protein shapes. Thousands, maybe even millions.
It'd be like having a 3D modeling software just for proteins.
Exactly. And think about what that could mean for drug discovery. Finding those elusive binding sites on a protein where a drug could attach.
So more targeted therapies, fewer side effects.
Potentially. And because SLM is so fast, it could speed up the whole drug development process.
So instead of years of research, we could potentially shave it down significantly.
Exactly. But drug discovery is just the beginning. This paper also dives into the potential of SLM for protein design.
Oh, protein design. Now we're talking about engineering proteins with specific functions, right?
You got it. It's like creating tiny molecular machines.
Whoa. So instead of just understanding existing proteins, we're talking about designing them from scratch.
In some cases, yeah. Or modifying existing ones, tweaking them to do what we want.
Okay, give me some examples. What kind of crazy stuff could we do with protein design?
Well, imagine enzymes that could gobble up pollutants, like plastic waste.
Whoa, eco-friendly enzymes. That'd be incredible.
Right. Or how about biomaterials with unique properties? Things we could use for construction or manufacturing.
So we're talking about a whole new era of bioengineering, where we can design proteins to solve specific problems.
Precisely. And SLM could be a key player in making this happen.
It's mind-blowing, but are there any downsides to this? Any challenges or limitations?
Of course, every groundbreaking technology has its kinks.
So what are we up against here?
Well, for one, we always need more data. The more data SLM has, the better it gets at predicting those protein structures.
So it's a hungry algorithm, always looking for more to learn from.
Exactly. And another challenge is making sure those designed proteins actually work, that they fold correctly and do what we intended.
Right. It's one thing to design it on a computer, but it's got to work in the real world, too.
Absolutely. So it's a constant back and forth between the computer model and testing it out in the lab.
Makes sense. So, big picture, why should all this matter to our listeners? What's the takeaway message?
This paper really showcases the power of thinking outside the box, using tools from one field, like language modeling, to solve problems in another, like protein research.
It's like connecting those unexpected dots and suddenly, boom, new discoveries.
Exactly. And this particular connection could change how we understand, study, and even engineer the very building blocks of life.
That's a pretty powerful thought.
It is. But hold on to that thought, because we'll be back soon to dive even deeper into the potential of SLM and what this could mean for the future of medicine and beyond.
Can't wait. This is just the tip of the iceberg.
And we're back, still geeking out over SLM and all its potential.
Right. I mean, we just scratched the surface of what this could mean for drug discovery.
Yeah, imagine actually designing a drug that perfectly hits its target, like a bullseye on a protein involved in a disease. Cancer, for example.
That's the dream, right? Targeted therapies with way fewer side effects.
Exactly. But how would SLM help us get there?
Well, each protein has these binding sites, right? Think of them like little pockets on its surface.
Okay, I'm picturing it.
And those pockets, that's where other molecules can latch on.
Like a drug molecule.
Exactly. But knowing the precise shape and location of those binding sites, that's critical.
Because the drug needs to fit perfectly.
Exactly. It's like finding the right key for a lock. The drug molecule has to have that exact shape to fit and interact with the protein.
Ah, the key to unlocking a cure.
Precisely. And that's where SLM shines. It can predict that 3D protein structure so accurately that we can see those binding sites in incredible detail. Way better than traditional methods.
So we get a high-resolution map of the protein, highlighting those crucial binding sites.
Right. So researchers can design drugs tailored to fit those sites perfectly.
Okay, I see where this is going. More effective drugs, fewer side effects, because we're hitting the right target every time.
Exactly. And remember how we talked about SLM being way faster?
Oh yeah, the protein research superhighway.
That speed translates to faster drug discovery, too. Think about it. Instead of years trying to figure out a protein structure and its binding sites.
We could potentially do it in a fraction of the time.
Exactly. That's a game changer for developing new treatments.
It really is. But we touched on this earlier. SLM isn't just about drug discovery, right?
Oh, no way. There's this whole other world of protein design.
Right. Remind me again what that entails.
It's like being a molecular architect. We're talking about engineering proteins with specific functions, like creating tiny, incredibly precise machines.
Whoa. So we're not just understanding existing proteins, we're designing completely new ones.
Sometimes, yeah. Or taking existing ones and tweaking them, modifying their structure or function.
So we're like protein hackers.
Haha, kind of. And SLM can help us do both, design new proteins from scratch or modify existing ones.
Okay, I'm on the edge of my seat here. Give me some examples. What kind of crazy stuff could we design?
Well, how about enzymes that can devour pollutants? Like literally break down plastic waste into harmless stuff.
Wait, seriously? That's amazing. We could clean up the environment with engineered enzymes.
That's the idea. Or how about biomaterials with crazy new properties, stuff we could use for construction, manufacturing, you name it.
So stronger, lighter, more sustainable materials.
Exactly. We could even design proteins that act as targeted drug delivery systems.
Whoa, like little protein couriers delivering medicine directly to diseased cells.
Precisely. The possibilities are mind-boggling.
It's like a whole new era of bioengineering.
And SLM could be the key that unlocks it all.
I'm totally with you. But even with all this potential, there have got to be some challenges, right?
Of course. Every new technology comes with its own set of hurdles.
So what are we up against with SLM and protein design?
Well, data is always a big one. The more protein data SLM has to learn from, the better its predictions and designs will be.
So it's a constant hunger for information. More data equals better results.
Exactly. And then there's the challenge of making sure our designs actually work in the real world. It's one thing to create a protein on a computer, but it has to fold correctly and function as intended in a living cell or organism.
Right, it's got to pass a reality test.
Exactly. So we need to rigorously test these designs in the lab to make sure they're stable and functional.
So it's a constant back and forth, a dance between the computer model and real-world experiment.
Right. We need both to make sure we're on the right track.
Makes sense. But let's zoom out for a second. For our listeners who might be thinking, okay, this is cool, but why should I care? What's the big takeaway here?
I think this research shows us the incredible power of AI and machine learning in biology. We're not just passively observing life anymore. We're learning to engineer it, to shape it in ways we never could before.
And that has the potential to revolutionize everything, from medicine to materials to the environment.
It's a reminder that we're living in an era of unprecedented scientific breakthroughs. Things that were once considered science fiction are becoming reality.
And as we explore these frontiers of protein science, SLM is going to be a crucial tool.
It's a journey full of possibilities, and we're just taking the first steps.
Welcome back. We've covered a lot of ground with SLM. We've seen how it works, how accurate and fast it is, and all that potential for drug discovery and protein design.
Yeah, it's been a wild ride.
It has, but now I want to zoom out a little bit. Look at the bigger picture. What does all this mean for society?
Well, this research is like a flag in the ground, you know. It shows how much AI and machine learning are changing things, not just in biology, but everywhere.
Right. We're using these tools to analyze mountains of data, make predictions, even design experiments. It's like sci-fi stuff becoming reality.
Exactly. And it's happening so fast, it feels like we're just at the beginning.
Exactly. But with all this power comes big questions, right?
Yeah.
How will SLM and protein engineering change our lives? What are the societal impacts?
Well, one huge area is personalized medicine. Imagine getting therapies designed just for you based on your genes and proteins.
Wow, that's incredible. It'd be like, goodbye, one-size-fits-all medicine. Hello, personalized treatments.
Exactly. And it's not just about treating diseases. Think about sustainability. We talked about those enzymes that can break down pollutants.
Yeah, eco-friendly enzymes. What else?
We could engineer biomaterials that are way more sustainable than what we use now. Less reliance on fossil fuels, less impact on the planet.
So SLM could help us tackle some of the biggest challenges we face. Disease, climate change, pollution. It's pretty mind-blowing.
It is, but you know, it's like they say, with great power comes great responsibility.
Right, there's always another side to the coin. What kind of ethical considerations are we talking about?
Well, any technology can be misused, right?
Yeah.
We have to make sure SLM is used ethically. Make sure everyone benefits from it, not just a select few. We need open discussions and clear guidelines.
That's a crucial point. We can't let the excitement of scientific progress overshadow the ethical side of things. We have to make sure we're using these tools wisely and responsibly.
Absolutely. Open dialogue, collaboration, and a commitment to ethical principles, those are essential.
Couldn't agree more. This deep dive into SLM has been an incredible journey. We've seen how it's changing our understanding of proteins, how it's accelerating drug discovery, and how it's opening up a whole new world of possibilities for protein design.
Yeah, it's been a real eye-opener.
It has. But for me, the most inspiring part is that it shows the power of human ingenuity.
Yeah.
Our ability to ask questions, to explore, to solve problems. And that's something we should all celebrate.
Absolutely. And who knows what other amazing discoveries are waiting for us just around the corner.
That's the exciting part. The future of science is wide open. So, to everyone listening, stay curious, keep exploring, and never stop asking questions. Because the pursuit of knowledge, that's what drives us forward, and that's what makes all this research so incredibly exciting. Thanks for joining us on this deep dive into the world of SLM.